Hello, welcome to my channel Biographia. My name is Shayan, and in this channel I aim to cover the various topics and chapters of CBSE and ICSE biology. So please stay tuned as I will slowly upload videos that will cover the entire syllabus across various standards. Hello, welcome back to my channel Biographia. In this video I will explain the structure of nucleus, DNA and chromosomes. It's not a separate chapter, it's part of the chapter Cell, the Unit of Life, which is a common chapter in both class 9 and class 10 syllabus. But to understand the structure of a cell, we need to understand the structure of one of its cell organelle, which is the nucleus, and what are the components that are inside a nucleus. So, to run through the basics, we have a cell. The cell is bound by an outermost membrane called cell membrane, and if it is a plant cell, it will have another membrane on top of this called the cell wall. Inside the cell, there are various membrane bound structures called cell organelles. What are cell organelles? These are living membrane bound parts of a cell that perform various functions of the cell. Of these various cell organelles, we have a special cell organelle known as the nucleus. And the nucleus contains thin thread-like structures called chromatin. And other rounded objects called nucleosome. So in this video our main focus will be on, not a nucleosome, nucleolus. In this video our main focus will be on this structure called chromatins. What are chromatins? So chromatin is made up of a macromolecule. What is a macromolecule? A complex molecular structure. So chromatin is made of a complex molecule called DNA. So that brings us to the structure of DNA. And how this DNA forms chromatins and chromosomes. So DNA has a single unit called a nucleotide, which in turn is made up of a variety of molecules which are as follows. So first, a DNA molecule, a single unit of DNA molecule is called a nucleotide. The nucleotide is made of a molecule called nucleoside and phosphate. Formula for phosphate, PO4 3-. Nucleoside is made up of a pentose sugar and nitrogenous bases. And there are two types of nitrogen bases: purines and pyrimidine. There are several types of purine pyrimidines, of which we are going to study the basic ones. Adenine, guanine, thymine and cytosine. So now let us move to the actual chemical structure of this. So let us start with the pentose sugar. So pentose sugar, the name comes from its pentagonal structure. What is a pentagon? A five-sided geometrical structure. It has 5 sides. Of these 5 sides, there is an oxygen at the top and 5 carbon atoms. So this is a pentose sugar, a pentagonal carbon ring with 1 oxygen. If we add a nitrogen base to this pentose sugar, we get a structure known as nucleoside. If we add a phosphate group to it, This forms a basic unit of DNA called nucleotide. What about the various types of nitrogenous bases? Nitrogenous bases, I am drawing it as a simplified box, but purines are double ring structures. I won't go to the details of this because it is for class 11 and 12. For class 10 level, just know that purines are double ring structures and pyrimidines are single rings. So this is one ring. If it has another structure, it is called a double ring structure. So I'm drawing the basics. Visuals are always better to understand something. At every corner there is a hydrogen, a carbon and other elements like nitrogen. So this is a double ring purine structure and this is a single ring pyrimidine structure. So can you find a similarity between the pentose sugar and this? A pentose sugar is a pentagonal carbon ring, while pyrimidines, it is a hexagonal carbon ring, and purines is a combination of a hexagonal carbon ring and a pentagonal carbon ring. So purines are A, G, thymines are pyrimidines are T and C, thymine and cytosine. So there is a tendency of formation of a hydrogen bond between adenine and thymine, guanine and cytosine. I will come to that later when I will draw the structure of a DNA, but just for basic knowledge, know that adenine and thymine form a double hydrogen bond between their structures, and guanine and cytosine similarly form a triple hydrogen bond between themselves. So this is the basic unit of a DNA, a nucleotide. Now, what is the full form of a DNA? It is deoxyribonucleic acid. There is another form of nucleotide which is also very common to biology chapters, so I am giving you a brief of it. It's called RNA. The full form of RNA is ribonucleic acid. So DNA is basically a ribonucleic acid, but it has one less oxygen molecule in it. That's why it is called deoxyribonucleic acid and RNA is called ribonucleic acid. So if we go to the ring structure of a nucleotide, a DNA nucleotide has a pentagonal ring. An RNA nucleotide also has a pentagonal ring. There is oxygen at the top of the ring and five carbon atoms, 4 inside and 1 outside the ring. So up till now, the structure of a DNA and RNA nucleotide is same. Then what's the difference? Carbon has a valency of 4, that is it can form 4 bonds. Carbon can form 4 bonds. So here each carbon has already formed 2 bonds. For example, we generally have to give numbers to these carbons. So it has carbon 1, 2, 3, 4 and 5. Similarly, here carbon number 1 has already formed 1 bond with oxygen and 1 bond with the second carbon. The second carbon has formed 1 bond with the carbon 1 and 1 bond with the carbon 3. So similarly, carbon 3 has formed 1 bond with carbon 2 and 1 bond with carbon 4. Carbon 4 has formed 3 bonds with oxygen, the third carbon and the fifth carbon. So there are extra hands that the carbon can use to form other bonds. So here it has 2 other free hands that join to a hydrogen and hydroxide group, OH group, hydroxyl group. So a DNA and RNA has its differences to the side groups that are attached to the second and third carbon. So in ribonucleic acid, one of the groups of the second and third carbon is OH. So one OH group is attached to the second carbon, another OH group is attached to the third carbon. But in DNA, the third carbon has an OH group, but the second carbon has an H group, which can be interpreted as a ribonucleic acid, an RNA, which has lost one of its oxygen from OH group, leading to the formation of a single H atom at the second carbon. So that's why deoxyribonucleic acid. Deoxy means subtraction of one oxygen from a ribonucleic acid molecule. So that's the basic difference between a DNA and RNA. There are few other differences, which are a DNA always forms a twisted double-stranded structure, hence it is called a double helix. And RNA is always a single-stranded structure. There are instances where RNA forms double-stranded helix, but it is very rare. But it is very common in DNA. DNA generally tries to form double helical structures or double helix structure, which gives it a better stability. The next difference between a DNA and RNA is the nitrogenous bases. DNA has purines, adenine and guanine bonding with pyrimidines, thymine and cytosine. But in case of RNA, the nitrogen bases are slightly different. In purine, it has adenine and guanine, but in pyrimidine, RNA have another type of nitrogen base called U, which is uracil. and G pairs with C, cytosine. So everything is same in the nitrogenous bases with only one change. In DNA, adenine will form two hydrogen bonds with thymine. So the nitrogen base thymine is present in only in DNA, but in RNA we don't have thymine. We have another pyrimidine called uracil, which takes up the place of thymine and bonds with adenine. So I will wipe the board now. If you want to take notes, you can take a screenshot. Now, how does this single nucleotide form a double helical DNA structure? I would draw three nucleotides on each strand to explain the double helical structure. So this is one pentagon of a DNA molecule. So these are the single nucleotide units. These nucleotide units now join with one another. How do they join? The third carbon of one nucleotide forms a bond with the phosphate group attached to the fifth carbon of the adjacent nucleotide. In this way, the nucleotide molecules attach to one another. Here more nucleotides are added and this part is called a polynucleotide chain. Poly means many. So a DNA molecule is actually a polynucleotide, double polynucleotide chains. So here we have many nucleotide units that form a long chain. Now what about the next one? The next one is similar to this but in a reverse direction. Similarly, like in this chain, here also the third carbon forms a bond with the phosphate of the fifth carbon of the adjacent nucleotide. The structure is very similar but in a different direction. Hence, DNA is called antiparallel. Antiparallel strands. DNA is made of antiparallel strands. So we may draw DNA like this, but they are parallel in nature. That is, the two strands will not at any point touch each other. They will wrap around one another like this, but they won't touch. So one of the strands is called a 3 to 5 strand because it starts with a third carbon and ends with a fifth carbon, and the other strand is called a 5 to 3 antiparallel strand. It starts with the 5 and ends with the 3. So this is the basic structure of a DNA molecule. There are many more nucleotides. The human cell, each nucleus of a human cell, each cell has about 3 billion base pairs. So what is the meaning of this? 3 billion is 300 crore. That is 9 zeros. 9 zeros. 9 zeros after 3. And base pair. What is a base pair? So here the nitrogen bases, which can be adenine, guanine. For example, this can be, say, adenine. So if this is adenine, the next one will be automatically thymine, because adenine pairs with thymine, guanine pairs with cytosine. So if this is C, this will be automatically G. If this is T, this will be A. So hydrogen bonds form between these nitrogenous bases, and each of these pairs of nitrogenous bases of the two strands of DNA is called a base pair. So our one human cell has a nucleus which consists of 3 billion such base pairs. So the total number of nucleotides in a human body, we can't measure it. Each single cell has 3 billion of these. So there are 3 billion base pairs. So you can imagine the length of a DNA molecule. The human cell has 46 chromosomes. That is 46 such DNA molecules. If I draw this as two strands, we have 46 of these double-stranded DNA molecule, and each DNA molecule has single base pair units called BP, abbreviated as BP, not blood pressure, base pairs. So across 46 chromosomes we have 3 billion of these base pairs. So it is really a long length of DNA in each human cell. So how do we accommodate such a long length of DNA in such a small space inside the nucleus of a cell? For that we need some packing mechanism. We would need to pack these DNA threads to accommodate all this into this small nucleus. So how does that packing begin? DNA is a double-stranded structure, but for better understanding, I will draw this double strand as a single one. So first, DNA I am writing is that DS means double strand. The DNA double strand folds, and to fold it requires the help of protein molecules called histones. So a single histone core molecule is made of 8 individual histone proteins. So 8 histone proteins make a histone core molecule. So this circle has 8 histone protein molecules inside of it. Now DNA wraps around this histone core, and this arrangement is called a nucleosome. So one histone core made of 8 histone proteins with a length of DNA wrapped around it in turns is called a nucleosome. And how does the DNA wraps around the histone protein? So histone is made up of positively charged proteins. So the histone core has a slight positive charge. And the DNA, since it has negatively charged phosphate groups, so P here is actually a phosphate group, PO4 3 minus. So DNA has a slight negative charged around it. So as we all know, positive charges attract negative charges. So DNA ultimately wraps around this histone protein molecule. Now DNA has wrapped around the histone. Then what? Then this will again, so if I consider this as a green thread, this will again coil. So to pack DNA into the nucleus, we have to supercoil the DNA to accommodate it inside the nucleus. It is again coiled. So if we consider this as a single thread, this will again coil around itself to form a supercoil. This stage is called a chromatin. That are the threads inside a nucleus. So there is another fact that we need to keep in mind. When a cell is in its normal stage, that is, it's normally doing its daily processes, it is not dividing. In that stage, DNA is found inside the nucleus as chromatin fibers. Chromatin fibers are only visible under a electron microscope. We can't see chromatins inside a nucleus with a normal compound microscope. And when the cell starts to divide, the DNA is further coiled to form even more condensed structures called chromosomes. Chromosomes are visible under a compound microscope. Since the chromatin further coils to form a chromosome, the size increases, the size of the structure increases, and it becomes visible under a compound microscope. We know the difference between a compound microscope and electron microscope. I will link the video where I have explained the differences between a compound microscope and an electron microscope. So a compound microscope has lesser magnification power than an electron microscope. Since chromatin coils to form chromosome, the size of chromosome is much larger than chromatin, it is thicker. So it becomes visible under the low magnification power of compound microscope. But when it is in chromatin stage, it is so thin that a compound microscope is not able to zoom in and focus on this structure. So to view chromatin fibers, we need an electron microscope which has even more magnification power. Now, the chromosomes only form when the cell is about to divide. So when the cell is not dividing, it is in its resting stage, like it's doing its normal day-to-day activities. So in that stage, DNA is present in the form of chromatin fibers. So since chromatin is less coiled than chromosome, the DNA in chromatin is accessible. If I now consider this chromatin as one thread, this will coil and supercoil. There are various other layers of coiling and supercoiling. I am skipping few parts because we don't need that in class 10 syllabus. So after several layers of coiling and supercoiling, what we get is a chromosome. A chromosome has two parts, a shorter arm two longer arms and a constricted region in the center called centromere. So that is the structure of a chromosome. So now, why does it has several layers of coiling? When the cell is in resting stage, DNA is like the code of our body. So like a computer program or a mobile app has a coding. Software engineers write codes that enable that software to run. Similarly, our human body has a coding of its own, and that is the coding which is present inside the DNA. So DNA contains the code of life. DNA contains the code that will direct our body to make whatever is needed by our body to live a normal life. So for example, I am eating a burger, so I am eating carbohydrates. So our body needs enzymes to digest that carbohydrate. Where will these enzymes be made from? That code is present in DNA. Whatever the body needs, everything, the code is present in our DNA. DNA holds that code. So when I need a carbohydrate digesting enzyme, our body will go to the specific part of DNA which has that code and will start making the enzyme from that section of DNA. So when a cell is not dividing, it means that it is doing its normal work. At that time, our DNA has to be more accessible. So if, for example, the DNA has certain segments from which we make these enzymes and other stuff that our body needs. If this is supercoiled and it gets buried inside the chromosome, we can't access it. If we can't reach to that part, how will we make that product that we need? So when the cell is its resting stage, the DNA is more free in the form of chromatin fibers, so that our body can access those segments of DNA and make whatever it needs. But when the cell goes into its division cycle, then the DNA is wrapped, because in the division cycle the only focus is to make 2 copies of each DNA molecule and make 2 cells of one cell from one cell. So that is why the normal functioning stops. So then DNA is again coiled to form chromosomes. So what are these sections of DNA called, from which various products are made? These are called, I'm making a separate diagram for this, otherwise you will be confused. So if this is a double-stranded DNA, segments of this DNA help us or hold the code to make products that our body needs. So these segments are called genes. So what is the definition of a gene? Specific sequence of nucleotide that code for a particular protein or product. So this is the basic structure of a DNA and how it is accommodated inside the nucleus of a cell. If you want to take notes, I am pausing for a second. You can take a screenshot. If you like this video, please subscribe to my channel and hit the like button. And if you have any questions regarding this topic, please drop your questions in the comment box. I will try to address them as soon as possible. Thank you. If you find my videos helpful, please like the videos and subscribe to my channel. And if you have any queries or you need clarification with any topic, if you have any form of request, please drop a comment so that I can help you in any possible way. Thank you.